Hello, and good day once again for the class. This time is going to be for general biology 2. Allow me to share my screen. Sorry, let me just stop share for a while. Okay, try if I can do this. Let's try this one. All right. So last time when you were still in class, we were talking about blood time. So just to review, you have A, B, O, and A, B. I asked you, I think you have time right now to have your blood types checked in the clinics that you are in. So I hope you can also do that. And hopefully you can also verify the blood types of your parents as well. Okay, for now, just have that here, keep that in mind, and then now we will go with our next topic, and that is about Mendelian genetics. Okay, so let's start. Let me just make this big. There we go. Okay. Gregor Johann Mendel, 1822 to 1884. He is known as the father of modern genetics. And unlike typical scientists that we know, Gregor Mendel is actually a monk. The modern concepts of genetics took birth from his pioneering. He works on, okay, what did he work on? That's the scientific name, Pisum satium, or a garden tea, from the periods of 1856 and 1864. Now, this is a fair warning for you that for Mendelian genetics, there are going to be so much terminologies that you need to remember. So I think for this one, it is best for you to take notes of the discussion. And of course, I'm going to be sharing the PowerPoint as well. Okay, there are also other sharings in this PPT, so I will just go over the things that I am familiar with and the focus of our discussion. Okay, scrolling down, Gregor Mendel did an experiment in which he crossbred pea plants, and that's a key term again, crossbreeding pea plants, and discovered that the traits could be passed on to another. Mendel's discovery with the peas is now used as a model system for primarily for inheritance. A model system is convenient because it may tell us how other organisms, including us humans, as I mentioned, actually function. Okay, and together with your blood type, it's gonna be, I would say, interesting and intriguing. Knowing your blood types. Okay, Gregor Mendel conducted heredity experiments, and we're going to be defining heredity later on. Okay, using common garden tea plants. Mendel cross-mated large numbers of tea plants. Mendel concluded that there were traits that always appeared or expressed when they were present in an organism. And how do we define that one? If that is expressed or present in an organism. Okay, so this is what he did. Okay, as an overview. Purebred plants are called parent P, signified as letter P, as you can see in the drawing, parental generation. And each generation would be represented with F. Okay, which represents first filial F1 generation. If there is F1, there is also F2, so on and so forth. Okay, now take note of the code. SS, what does that mean? There is two double S here and two small double S. Okay. X would represent the crossbreeding. Okay. The trait that always appears when it is present is called a dominant trait. Okay. If it, as defined in here, if it is always present, that it is dominant. Now, notice on the drawing that the parents having small capital S crossed with small letter SS produces gametes. And for gametes, usually if you have two genes over here, you would have to produce two, two traits being divided. So there's going to be another S here and SS here. We will be discussing on this one when we go through hybrid process. In particular, we'll just focus with mono, and I think we can try to go with dihybrid cross, which is going to be also interesting. The trait that always appears when it is present is called a dominant trait. Okay, take note of that one. P for parent, F1 for the first generation or first filial generation, and dominant trait as a definition is out already. Next, the trait that is hidden by the dominant trait is called the recessive. So the traits they define this as traits. Later on, you would see that they also call this as a gene. Okay, a dominant trait and a recessive. How do we define recessive trait? The trait that is hidden by the dominant that is called the recessive. Now, remember that the terminologies would use present, and then the other one is hidden. It doesn't say that it's gone, it's just hidden. And take note of that one. Okay, next slide. Mendel's genetics. Mendel's pea plants were true breeding. And how do we find true breeding? Meaning that if they were allowed to self-pollinate, they would produce offspring identical to themselves. Okay? Say, for example, if you look at the picture, if this papaya would produce yellow, ripe papaya, then definitely its offsprings would also produce a true breeding, which would also have a yellow ripe papaya. Okay? Mendel wanted to form different pea plants other than his true breeding plants. So he cross-pollinated. Okay, now there is the true breeding, another terminology, and cross-pollination. Cross-pollination, if this one would produce just two orange, then if you're going to cross-pollinate, then definitely there is going to be another trait that is involved on this one. Okay? He cross-pollinated his pea plants by joining male and female reproductive cells from different plants. Okay, this is just an example, but of course, with Mendel's genetics, he was practically using a pea plant. Okay, the role of fertilization. Here comes the term again. Fertilization during this process is when the male and female reproductive or male and female reproductive cells would be joining together. Okay. Of course, we are commonly familiar with how a sperm and an egg would produce sperm for male and egg for female. And if they are joined together, that process is called fertilization. The one that is formed, what do we call that one? If you can still remember, meaning it is a fertilized egg already. Okay, and it starts with letter Z. Who can remember that word? Okay, that's a zygote. Okay, trait is a specific characteristics. For example, the color of the flower. Now, um, they call it trait which is actually the one that is expressed. But what about the one that we're talking about, the one that is hidden? Okay, take note of that one. When Mendel began his experiment, he used the pollen to crossbreed them. He knew, sorry for that one. He knew a male reproductive cell, which was a sperm, and a female reproductive cell was an egg. That's the terminology there. Okay, next. Okay. Then here goes the cross pollination involving, okay, of course, you have a male and a female with different traits. Okay, Mendel studied seven pea plant traits, which will be tackling later on. There are seven, but we will not go through some of the traits that he's going to be doing. Okay, trait is a specific characteristic such as seed color or plant height that varies from one individual, one pea plant to another. Hybrids are the offspring of process between parents with different traits. Okay, we have done this one or P for parental generation, and if you have F1 for the first generation of the sons and daughters of your parental generation. Next slide. Okay, this is just an illustration of how we did it. Okay, no need to go over this one, but yes, you can refer to this one and go over with this slide. Next, these are the characteristics now or traits. Okay, take note of this one. Seed shape. There will be representation on this one. Okay, which will be discussed on another video. Seed shape. But for this instance, you can refer to this. Either we would represent the seed shape. If it's round, then how do we represent that? Are we going to represent it with letter R, or are we going to represent it with a W, which is wrinkled? Now, there are two traits that you need to remember. The traits for genes could actually be dominant and recessive. Seed color, yellow or green. Seed coat, it could be gray and it could be white. The pod shape, it could be smooth and constricted. Pod color, green or yellow. The flower position, eggshell and terminal. And the plant height, either tall or short. Okay? And this is just an example of their F1, the first generation, having round, yellow, gray, smooth green, eggshell, and tall. Now, if these are the traits that are coming out, how do we define the other traits then? Okay, now take note of the dominant and recessive traits. Which do you think is dominant and which do you think is recessive? Having the F1 having round, yellow, gray, smooth, green, eggshell, and tall. Okay, so it is not a hypothesis that you're going to be making, but this is an obvious answer of what happened to the other traits and how would you define that the other traits that were not shown or there were that did not that were not expressed during the F1 stage. Okay, let's move on. Pea plants have several advantages for genetics, and these are the reasons why Mendel used this one. Pea plants are available in many varieties with distinct heritable features or characteristics with different variants or traits. Another advantage of peas is that Mendel had strict control over which plants mated with which. Each pea plant has male stamens and female carpels or sexual organ. Now you need to know the parts of our plants then. We will have to go through that, and the best example that we have would be using the flower of the goma mela. Now hopefully we can explore that one and do an experiment on that one. Okay, and really watch it under the microscope and how do they look like. The pollen grains, you're gonna be seeing that, and the eggs or the ovums or the eggs of the gomamela. Now, hopefully, we could see that one also under the microscope. In nature, pea plants typically self-fertilize, okay, fertilizing ova with their own sperm. However, Mendel could also move pollen from one plant to another. This is what he did, as shown in there. He manipulated whatever he could manipulate to produce a different variety or different and hybrid pea plant. Okay, in typical breeding experiment, Mendel would cross-pollinate, hybridize, another terminology, cross-pollination or hybridization or hybridize. Two contrasting true breeding pea plants varieties. The true breeding parents are the pea, again, once again, and the F1 or the hybrid offspring are the F1 genetic trait. Mendel would then allow the F1 hybrids to self-pollinate to produce an F2. And this is the process that he went through. We will not go through this one. Let's skip to that one. And these are his major conclusions. First, sorry, excuse me. First, was that biological inheritance is determined by factors that are passed from one generation to the next. And scientists call this gene, genes. Second, principle of dominance. Now, as we mentioned, there is a dominant and recessive trait. States that some alleles are dominant and others are also recessive. And take note, alleles is out already, and there is going to be a definition of what alleles are. And the third is during gamete formation. I hope you are familiar with this one, gamete formation, alleles segregate from each other so that each gamete or sex cells carries only a single copy of each gene. Now remember, they always come in pairs, but during gamete formation, that's the making of their sex cells, either it's a sperm cell or an egg cell, those traits are always separated and a single copy. Each F1 plant produces two types of gametes. Those with allele for tallness, for this trait itself, either tallness or shortness. It could also be tallness, tall, and it could also be short. And there is another definition of that one. If they have both of the genes would carry too tall or too short. Okay, scroll down. Review discovery of Mendel's original work. In the 1900s, there are other scientists. Okay, Carl Correns, Hugo de Vries, and Eric von. This is a mouthful. Chesermont. Okay, we're now known as, what do we call it? Mendelism or Mendelian, not laws. This should be LAW, laws of inheritance. Sorry for the typo. Okay. Now here we go with the terminologies. Okay. Coin. This is a very good term. I love this word. Coin, which means that person made that word. Okay. The term genetics was coined by William Bateson. The term gene was coined by Joe Hansen, 1909. The definition is a gene is the hereditary determining factor. Okay. Gene consists of a continuous segment of DNA, as we all know. And this term, locus or loci, what does that mean? Okay. Sorry, let me just scroll a bit. And it would refer to a specific position, I would say a location on the chromosome. Okay, you can also see in here of how chromosome looks like. Let's go on the next slide. Take note of this one, locus or loci. Okay, a locus singular and loci would be plural. Okay, continue. Okay, here goes. Allele. Or the other, you can also call it as allelomorphs. I would just commonly call it allele or alleles. What is the definition? Are alternate forms of a gene which occupy identical loci. Loci, we discovered already that this is the location of that gene. Okay, on a homologous chromosome. Homologous, they similar. Okay, allele control the contrasting characteristics of same trait. As we mentioned, usually alleles exist in two different forms. Now this is clear. Um, these are basically chromatids and what you see in here, but if they're put together, we call them as a chromosome. And each segments of these chromosome, for this instance, chromatid would actually carry a single gene or a single trait. Okay. Now, this trait, for instance, basically occupies the locus for the flower color. The locus or the location of the, what is that one? Okay, flower color of the gene. They should be on the same position. Okay? And what do we call that position in there? You call that the allele. The position would be the locus or loci. Homologous pair of chromosome. Okay? These segments, okay, or loci, this is the location, would carry what? Okay, a specific gene or a specific trait. And for this instance, this is the flower color. Okay, so take note of the terminologies. Okay, let's move on. Okay, dominant and recessive alleles. Dominant allele will always express. Okay, uh, of course, coming from the term they're dominant, so they would always be the strongest. They always show or express whatever they carry in there. Recessive, however, is would mean the opposite. Okay, will express only if there is no a dominant allele or a dominant trait. Dominant allele mask or suppresses the expression of a recessive allele. Now, does that mean that it deletes the recessive allele? It only, okay, suppresses, but the allele or the recessive allele is still there. Dominant alleles are classically symbolized with capital letters. Remember the SS and the small s? Okay. It should be expressed in capital letters. And the recessive alleles will be in small letters. So which one should we carry then? Okay, let's go. Now, in terms of height, you can have tall and dwarf. Now, considering the tall is actually what? The dominant trait. So we need to carry it as a letter T. Okay, we carry tall as the letter D as a dominant trait, while the small letter D would represent either short or a dwarf. Okay, as stated in this example. Okay, there are other instances. What happens? Okay, there are also multiple alleles as defined in here. Many genes exist in several different forms and are therefore said to have multiple alleles. A gene with two or more alleles. For instance, they are showing this as a rabbit's coat color is an example. The color of a rabbit's coat is determined by one gene with at least four different alleles. Now imagine that one. Commonly we only have two, but in the case of rabbits, they have multiple alleles. Next, okay, this is just a picture. Okay, another terminology, genotype and phenotype. If you just take geno, what would be the key term? Okay, or the keyword that you can relate it with? You relate it with the word genetics or gene. Okay, genotype would mean the genetic makeup or constitution of an organism, while phenotype would refer to the physical features or appearance of an organism. Okay, if I'm gonna ask genotype, we talk about the composition of the gene, and if we talk about phenotype, it only refers to the features. And soon, if we do the monohybrid cross, you will know the specifics on this one. But for now, Phenotype is produced not only by the genotype, but also the interaction between genotypes and environmental factors. Okay. For this instance, you have trait, height. The phenotype is tall and dwarf. Oh, sorry. Okay. The genotype is could be okay, tall, uh, two capital T's and a big letter and small letter T, or it could have just the small letter T's. There is going to be another terminology that you need to remember as we continue. Okay, other than just looking at the all capitals and a combination of a capital, small letter, and all small letters. Okay, here goes. Genotype is genetic, as mentioned, genetic makeup of an organism, and phenotype is the external appearance, what we could just see on the outside. And normally it is expressed by which trait? Is it a dominant or recessive? For an example, that looks tall can have a genotype that is pure tall or hybrid tall. And this is because when the dominant trait is present, that the organism expression shows the dominant trait. But if you see it as tall, does it mean that it has a pure trait or a pure gene of tall? It could actually be a combination of just pure and actually a combination of a capital T and a small letter T. Let's see if the terminology could come out on the next slide. Okay. This is given already. Genotype, genetic makeup, phenotype, it would mean the physical traits. Okay. And as given in here, just because something has the same phenotype does not mean it can have the same genotype. Exactly. Okay. Finally, it's here now. Okay. Um, Key terms, homozygous and heterozygous. Homo, what does that mean? A condition in which both members of an allelic pair, okay, the alleles, in the homologous chromosome are identical if it's the same or pure. Okay, in this case, Sol, TT, both capital letters are homozygous. It could also be dwarf or short, having small letters. You call that homozygous. If you talk about the phenotype on this one, this is tall, and for dwarf, this the phenotype would still be dwarf or short. Heterozygous, what does that mean? Okay, a condition in which the members of the pair, allelic pair, in the homologous chromosome are not identical. One is dominant and one is recessive. Now remember that dominant traits would always be expressed. Okay, if I'm going to ask you what is the genotype on this one, this is when this terminology would come out. Either you use homozygous or heterozygous. And coming from the example, what would be the genotype of this offspring? Is it homozygous or heterozygous? The genotype would mean that this is actually a heterozygous, having a small letter and a big letter and a small letter. Going back to here, okay, this is homozygous. This one would mean this is heterozygous, a combination of the dominant and recessive traits. Okay, these are the terms. You can go over this as I have defined already. And hemizygous, I am not commonly using this one, but it is a condition when genes are present only in one copy. What does that mean? Genes on X chromosome in male uh, have one X and one Y chromosome. Genes on Y chromosome only one Y chromosome in male. Okay? Usara ko na ikukya. Hemizygous. Okay. Dominance has been discussed already, and recessive has been discussed already. Okay? Dominant and recessive would mean the principle of dominance. dominance. Okay, moving on. Okay. Hybridization. These are just terminologies that you need to familiarize. The process of crossing two genetically different individuals. And in this case, you have a homozygous tall crossed by a homozygous dwarf. All capital T and two small t. Hybrid. What's the meaning? The progeny of hybridization is called a hybrid. This is now a basically a hybrid. We will be talking about monohybrid, which means that organism which is heterozygous with respect to only one pair of allele at a locus under a study. In this case, we will just be crossing one particular trait. Die from the keyword die would mean we will be crossing two traits for that matter. Okay, you will see in here uh, yellow round seed and green sprinkled seed. Monohybrid as defined and dihybrid as defined. All right. Let's scroll down. Okay, reciprocal. We'll just go with the terminology in this one. Okay, two reverse process in which the sexes of the parents are interchanged. Okay, autosomes. Okay, remember that one. Those are the body cells. Let's scroll down. Okay, back cross is just a terminology that we need to be familiar with. We will not be focusing this one much. Okay, test cross, another terminology. Okay, and you would see here just a picture of the principles of dominance. Okay, tall, short, all tall, of course, because the tall is dominant. All tall, full. Why is this short now? Okay, we will go through this later on if we do. Okay, there is also an incident called codominance. And what does that mean? The phenotypes produced by both alleles are clearly expressed. Okay, looking at the picture, there is red and there is white. This is not a pea plant, which is a certain plant having two colors. And that would mean that both of the traits, the red and the white, are actually dominant and are also expressed. Okay, codominance. Incomplete dominance. What does that mean? uh, Blending inheritance. Okay, which means that if we take, for example, in this sense, if we mix white and red, it produces pink. That's also a very nice uh, instance. Incomplete dominance, codominance. Okay, this is the codominance and the shared traits. Okay, what does that mean? These are the terminologies that we need to remember. Incomplete dominance, codominance, there is a multiple alleles and polygenic traits that we will not be talking so much. Okay, and I will be entertaining your questions as I see you. That's okay. And this is it. Let me just scroll back. And this is it for Mendelian Genetics. Okay, thank you and see you in class. Bye bye.